We've talked about DNA replication, and we've also talked about the fact that DNA contains genes to make proteins. So now we're going to specifically talk about the process of protein synthesis. Remember, synthesis means to create, so in this case we're talking about the creation of proteins. So protein synthesis is the process of reading the instructions in the DNA to make a polypeptide. Remember that a polypeptide, poly means many, pept or peptide is talking about a protein. So this is talking about a chain of amino acids. And that chain of amino acids can bond to other chains of amino acids to fold into a protein. So all of this falls under the central dogma. When you hear that term, just think about like an overview. So this is the overview of protein synthesis. I have DNA, which is the instructions that are located in the nucleus, which it can't leave. So this is an issue because proteins are made in ribosomes, which are not in the nucleus. So this means that protein synthesis has to take 2 steps. The first step is transcription, and that's when DNA is going to be made or going to be making RNA. And then the second step is translation, where that RNA is going to be literally translated into creating a protein. We're going to be discussing 3 types of RNA in our process here, so we want to briefly go over them. The first type is mRNA. The M stands for messenger, and this is going to create a copy of the instructions of DNA and carry it from the nucleus to the ribosomes that are in the cytoplasm or the ref-ER. Then you have tRNA, and the T stands for transfer. So this is going to bind and carry specific amino acids to the ribosome. So it's going to be transferring amino acids to the ribosome in order to build a protein. And then finally we have rRNA, which stands for ribosomal RNA. So this is what builds a ribosome along with proteins. And they also can help catalyze the formation of peptide bonds. So that means they can also help form the bonds that bond amino acids together to build the bigger proteins. So let's look at transcription. Its main purpose is to carry the code or the instructions out of the nucleus from the DNA. Because remember, DNA never leaves the nucleus because the nucleus protects the DNA. And proteins are made in the cytoplasm by ribosomes. So they are not located in the same place. I have to get my instructions from point A, the nucleus, to point B, the ribosomes. So transcription is going to happen in the nucleus, and it's going to start with DNA, and it's going to make mRNA, the messenger RNA. So how this is going to happen, RNA polymerase is going to bind to the DNA promoter where transcription is going to begin and unzip the gene that needs to be copied. Then that RNA polymerase is going to use my complementary base pairing rules to match RNA nucleotides with the exposed DNA nucleotides. This is very, very similar to the DNA replication process. So my DNA gets split open, RNA polymerase comes in and base pair matches just like it did similarly with DNA replication. However, remember, RNA does not have thymine. It has uracil. So adenine is going to bond with uracil instead of thymine, but cytosine still bonds with guanine. Once it finishes copying that gene, it's going to release the completed mRNA molecule, and then the DNA zips back up and the mRNA leaves the nucleus and enters the cytoplasm. So think of it like the DNA is a book, a textbook, and the RNA polymerase is just going to copy a chapter, aka a gene, out of the textbook. So you open up the textbook, you copy down the information that's in that chapter, and then you take the copy of notes with you, close the book, leave it there in the library. So here's an example of transcription. I have a DNA sequence that I'm using as a template, and it's in the 3' to 5' direction, and I have this sequence of nucleotide bases, TAC, GC, TAG, TAC, GATT. Okay? The mRNA sequence that's going to be made out of that is going to be anti-parallel, so it's going to be in the 5' to 3' direction because it's in the opposite direction as the DNA sequence. I'm going to use my base pairing rules, so T is going to be paired with A. A would normally be paired with T, but remember, mRNA does not have thymine, instead it has uracil. So the A pairs with U, C's still pair with G's, and G's still pair with C's. So I can go through and write my mRNA sequence using my base pairing rules. Moving on to translation, there are a few things that we need to refresh or know about our vocabulary. The first one is genetic code. So this is the code of instructions for how to make a protein. We're going to be using a codon chart in a minute to figure out which amino acids are going to be coded for with these specific nucleotide sets. So then you have a codon, which is a set of 3 nucleotides that's on the mRNA. You have an anticodon, which is going to be the complementary 3 nucleotides to a codon on the tRNA molecule. And then you have amino acids, which those are the monomers or the building blocks for making proteins and that are held together by peptide bonds. So a codon would be a set of 3 nucleotide bases on mRNA. So AUG here would be a codon. CGA would be a codon. UCA would be a codon. The purpose of translation is to read and follow the instructions that were carried by the mRNA to actually build the polypeptide, aka the protein. This is going to happen in the ribosomes, and remember that makes sense because ribosomes' purpose is to make proteins. It's going to start with the mRNA instructions and it's going to end with a polypeptide, a protein. So looking at it with a little bit more detail, the mRNA, so in my picture the mRNA is this red strand here that's been copied. It's going to attach to a small portion of the ribosome, and then the ribosome is going to read the mRNA in codons. So remember, that's in sets of 3 letters. It's always going to read it in the 5' to 3' direction, starting at the AUG codon. So remember, AUG is the start codon. Think about school typically starts in August. In August, shorthand, AUG. So one codon is 3 RNA nucleotides. So if I look here, AUG, here's my start codon right here. The tRNAs act like taxis and they pick up and drop off the amino acids that match with each codon. So this blue business here is the tRNA and the little black dots are the amino acids. So this tRNA has the anticodon UAC. And so the UAC is the anticodon that matches with AUG. That makes sense because of my base pairing rules. So this tRNA is going to come down and it's going to sit this amino acid down. And then the next tRNA molecule is going to come and it's going to pair its anticodon with the matching codon, and it's bringing an amino acid. And these amino acids are going to be bonded together with peptide bonds. And it's going to continue to go until something called a stop codon is reached. So when the mRNA has a stop codon, the tRNA knows to stop bringing amino acids and that the polypeptide chain, the protein chain, is completed. When we are talking about which amino acids are going to be brought with which codons, we have this genetic code here, which is called a codon chart. You don't have to memorize this codon chart, but you do need to know how to use it. So starting in the middle is going to be your first letter of your codon, and then you're going to work your way out. So let's say that the codon is the start codon, AUG. So I'm going to start in the middle with A, and then I'm going to move to the second ring and go to U, but the U that is specifically touching the A. So AU, and then work my way out, G. So AUG codes for methionine. Let's try another one. If the codon is UAU, what amino acid do you think it is? code for the amino acid tyrosine. What about GAC? GAC would code for the amino acid GAC, aspartic acid. So let's work through an example starting with transcription and ending through translation. So if my DNA sequence is 3' to 5', TAC, GCT, AGT, ACG, ATT, using my base pairing rules to create an mRNA sequence, I know it's anti-parallel, so it's going to be the 5' to 3' direction, AUG, CGA, UCA, UGC, UAA, because of my complementary pairing rules. So codons are just sections of 3 nucleotide bases in the mRNA sequence. So what I did is I took this and I just split it up into sets of 3 letters. And we know that AUG is always going to be the first one. So these are my codons. So I can use my codon chart to read these to figure it out. So AUG codes for methionine. So there's methionine. CGA stands for arginine. And it continued on until it got to UAA. So UAA is a stop codon. Since I hit the stop codon, I know that that polypeptide chain is done. And just for good measure, these would be the anticodons that are bringing these specific amino acids on the tRNA molecule. Remember, they're going to be the complementary pairs to the codons. So A pairs with U pairs with A, G pairs with C. So in summary, transcription and translation have similarities and differences to DNA replication, so we're kind of including it on this summary chart here. But the purpose of DNA replication is to make identical strands of DNA. I'm making copies of it. It's going to have the nucleotides adenine, thymine, cytosine, and guanine because we are dealing with DNA. And since we're dealing with DNA, it occurs in the nucleus. Transcription, on the other hand, its purpose is to produce mRNA from DNA so that the mRNA can leave the nucleus with the instructions. Because I'm dealing with RNA, my nucleotides are adenine, uracil, cytosine, and guanine. But because I'm dealing with DNA, it happens in the nucleus. And then translation, my goal is to actually build the protein from the instructions that I had in the mRNA. And I don't have specific bases here to work with. I'm using amino acids to build my protein. Since I'm building a protein, this is going to happen in the ribosomes because that's the ribosome's job, is to build proteins. This is a picture just showing a cell and overall where things happen. So here's my nucleus. Transcription is going to happen in the nucleus where I have DNA and an mRNA strand is being made from the DNA strand. So that mRNA strand is going to leave the nucleus and head to the ribosome that is outside of the nucleus in the cytoplasm, and that's where translation is going to happen. So I've got my amino acids that are getting picked up by my tRNA molecules that are coming in, matching with my codons and building a protein.